Hello students, once again welcome back to our biology class. Dr. Raghavendra here. In the earlier class we were discussing about the human genome project. We studied about basically the introduction, why it's called as mega project. We discussed what are the goals of the human genome project. We discussed history of the human genome project. Also we studied applications. We discussed. We were discussing about the methodologies. In the methodologies also we studied ESTs. What is the full form of EST? Yes, expressive sequence tags. So all the DNA that cannot be expressive as a RNA, only 2% of the DNA can be expressed as a RNA. So such expressive sequences of the DNA you need to identify, that is called as expressive sequence tags. And then we also studied about mainly what it is. It is called as the sequence annotation. Means the DNA has mainly the expressive sequences, we call them as a coding sequence. DNA has a non-expressive or non-coding sequence also we need to understand. And then the one more techniques, yes, one more what you can say the methodology is applied. What is that? The molecular cloning. What is that? Insertion of the human DNA into the plasmid. So make the bacterial artificial chromosome, and then yes, the bacterial cells keep on dividing, redividing, and they are multiplying. At the same time, the DNA what you have inserted human DNA into the plasmid, that also will get multiplied, or multiple copies we get. So these things we have studied, and then there are till more two important methods which have been utilized in the human genome project we need to study. What are those? We call it as the automated sequencing. Well, so automated sequencing based on the principles of Frederick Sanger. So Sanger's method we call it as, that has been very extensively used mainly in the sequencing. So how actually it works? So as such we know that the DNA is a double stranded structure. It has what we can say that adenine, yes, thymine, guanine, cytosine. So adenine always pair with the thymine, then the thymine pair with the adenine, guanine pair with the cytosine, cytosine will pair with the guanine. So this is the DNA as such. So I'm just going to give you the brief idea what is this automated sequencing. Yes, mainly by the Frederick Sanger's method. What they done? So when we add restriction endonuclease, what will happen? When we add the restriction endonuclease, it will cut the DNA. There are around 250 variety of bacteria, from which nearly around 900 types of restriction endonucleases can be isolated. So here the specific type of restriction endonuclease, what they will target certain repetitive sequences only. They will cut the DNA. So such a huge long lengthy DNA we don't want. We want the particular sequences or the fragments of DNA. What will happen now here? Once we add the restriction endonuclease, yes, the DNA will be now going to made into the fragments. So here the DNA, yes, as such fragments, and also yes, it is a single strand DNA. So to this we need to add particular kind of what we can say, yes, the single strand DNA. We need to make it as a hybrid DNA. So what we need to add? So we need to add dideoxy, yes, mainly adenosine triphosphate. Dideoxy, we can say that guanosine triphosphate. Dideoxy, we can say that cytosine triphosphate, CTP. Dideoxy, what we can say that thymidine triphosphate. What is this dideoxy? So these are deoxynucleotides. Yes, radio labeled. They are radio labeled. So what is this radio labeling? Mainly they are tagged with P32 or S35. These are the radioactive elements. What will happen now? They will form the DNA, yes, double stranded or hybrid DNA, it will form. And in this, what will be more? Yes, adenine, in this guanine, in this cytosine, in this thymine will be emitting the light. Yes. So when it is reflected, when it is exposed to the UV rays, it will emit the light. So now it becomes the radio labeled hybrid DNA. Hybrid DNA, it has been formed. So now what we are going to do, according to the, yes, Friedrich Sanger's method only, yes, we are going to do the gel electrophoresis. So in the gel electrophoresis, yes, there is, we can say that the wells, which are the cavities in which we are going to add the DNA sample. So by adding the DNA sample, yes, we are making the DNA to keep moving. So one, we are adding the thymidine here, this mixture, we are adding this cytosine mixture here, and then guanine, yes, here. So all 4 DNA are the same copy only. These all 4 are the same DNA only, but we can't make out the position of adenine, position of thymine, position of guanine just by seeing that. We need to make them as a hybrid DNA and then apply them for the gel electrophoresis now. Now what will happen? So here mainly the band will form. There are rest of the other bands also will form, but the radio label band will form here. Then yes, there is again a number of other bands will form, but here the radio labeled band is here. Then again in the third gel also, number of rest of the bands will form. Again some radio labeled band will form. It is a common same DNA, but the same what you can sequence they have. They are the duplicate of one another. They are the clone of each other. Okay. So only we have done the proper labeling here with the radio label. So here the bands will be there, but radio labeled band will come here. So what will identify? Yes, when it is mainly applied for the gel dock, it will say that adenine position in here, then it is the adenine. Yes, here it says that it is guanine position. Yes, it says mainly the cytosine position. It says here it is mainly thymine position, like this. Yes, automated machine sequencing will be done. We call it as the automated sequencing. Automated sequencing is done. So this is the sequence we get, like this. DNA isolated from various type of chromosomes can be automated sequencing is done, because millions of sequences are coming out, but the human manually cannot do all this. So automated sequencing will be done. We can call them as mainly the overlapping repetitive sequences are identified. Overlapping sequences identified. So thus the what we can say, when it is this gel sheet is inserted into the gel dock, we can say that now further what will happen, the scanner, so gel scanner will scan it, and then the information will come on the screen. So on the computer screen, yes, information will come. As such, it may be the adenine, guanine, cytosine, thymine. Yes, there may be the thymine, cytosine, guanine, adenine. Like this, sequences keep on coming, and those are stored in the, yes, mainly the memory, and that is called bioinformatics. This is what actually the things done in the DNA fingerprinting, we can say, or the Friedrich Sanger's method of DNA sequencing, what you are observing. So what they did, the Friedrich's method says that take the DNA, yes, isolated, yes, it is a huge mass and billions of sequences are there, but we are interested in a particular DNA sequences only. Yes, we are interested in the sequence of the insulin, we are interested in the sequence of the growth hormone. Yes, we want to know where actually they are located, particular chromosome, yes, particular chromosome sequence, particular Chromosome sequences are taken out only when you insert a specific restriction endonuclease. So restriction endonuclease are the enzymes. They cut the DNA at a particular sequence, and then what you get, one particular sequence, the enzyme you have used, all the DNA fragments are the same sequence, and they are made into the single strand also, and to that you are adding the radio labeled, okay, the complementary bases, dideoxynucleotide bases you are adding, then they form the base pairs, but particular base only will blink, or it will mainly emit the light when exposed to UV rays, and then yes, the gel is prepared. The gel is scanned by the gel doc scanner, and the information is stored in the computer. So this is a fast sequencing method that has been extensively utilized, and mainly based on the overlapping sequence analysis is done. So mainly done by the Frederick Sanger method. I hope this point or this method is very clear to you people, and this is what we say mainly the automated sequencing method based on the Frederick Sanger's method of sequencing. Well, let us understand there is one more method, what we call as the physical mapping. So we need to understand physical mapping also. Fine. What is this physical mapping? Means you are isolating the particular chromosome and staining the chromosome and localizing the particular genes on the chromosome. That is called as the physical mapping. So let us understand now one more method called as yes, physical mapping, which has been used in the Human Genome Project. So here the methodology in that physical mapping. What is this? Method of, it is the method of localizing specific gene on particular chromosome. Okay, so imagine here is a chromosome I have mentioned, and the chromosome has the gene A, gene B, and the gene C. So this is the chromatid arm and centromere, and then these are what we call them as loci. Loci means location of genes. Locus means single, genes means plural. So genes on chromosome based on the distance from the centromere. Okay, so this is what we can say physical mapping. By we also call it as the banding technique, staining or chromosome banding. Okay, we call it as. Thus physical mapping says that there are a variety of genes located on a number of what you can say locations. So if we consider the gene as such, so the genes identified based on satellite sequences. What are these satellite sequences? These are mainly repetitive, yes, sequences, sometimes referred as VNTRs. What is these VNTRs? VNTRs are variable number of tandem repeats. So these variable number of tandem repeats unique. So these are the unique. Okay, and varies person to person. person to person. So definitely 99% all the human DNA is the same, but the type of VNTR we are having different from one another. Okay, so these VNTRs are the most important site, or these VNTRs are helpful to understand even the genes. So the repetitive sequences, we call them as the satellite sequences. Satellite sequences, let me tell you here the simple example. Imagine here I am mentioning CCAA, CCAA, CCAA. So here what you are observing, these sequences are repeated. So on the DNA we are finding these sequences are repeated, and we call them as microsatellite sequences. Microsatellite sequences, because the repetitions can be in between 2 to 6 base pairs. So repetitions are 2 to 6 base pair repetitions. When the repetitions, yes, we call them the microsatellites, or it can be mini satellites also. Mini satellite sequences. Okay, so mainly the repetition are 10 to 60 bps base pairs. Okay, so thus by location mainly, yes, localizing what we can say the position of the gene, we call it as mainly, yes, it is a physical mapping or better banding or staining technique. And then the staining, yes, mainly you can say that location of a particular gene, why the gene are mainly identified, what is the basis of identification. When there is a lots of repetitive sequences, we call them as the satellite sequences. They are also known as the VNTRs, variable number of tandem repeats. See here, triple C and double A, triple C double A, triple C double A repeated. So these are called as mainly VNTRs, variable number of tandem repeats. The repetition is a very particular, or the pattern of repetition is very peculiar. It varies from person to person. So uniqueness is there in the repetition. So this can be useful in identifying the difference between the person to person, because 99.9% DNA same in all the human, but the fingerprinting technology utilizes the presence of these VNTRs. So VNTRs or the repetitive sequences, we can call them as satellite sequences, and these satellite sequences can be microsatellite as well as mini satellite. So mini satellite, why? Because the 10 to 60 base pairs repetition, where 2 to 6 base pair repetition is a microsatellite. So this is all about the methodologies what have been utilized mainly in the sequencing or sequence annotation, we can say, or physical mapping. Let us go ahead with the salient features of mainly Human Genome Project. So salient features also mainly very many various features, yes, we can find varying features we can find, but all the features can be well understood or well studied. So you need to remember lots of numerical information. So number based information, yes, you need to remember. That is what we say mainly, the because number based question also may come. That's why you need to remember these very well from the point of NEET exam as well as board, both important, yes. So let us go ahead with salient features of mainly the Human Genome Project. Salient features of Human Genome Project. What are those? Mainly, number one, yes, we can say the features can be categorized as nucleotide based features. As such, how many base pairs? So, approximately we can say that 3 into 10 raised to 9 base pairs. These many huge number of nucleotide, okay, base pairs we can find. And then, yes, other than that, we can say that 99.9%, yes, bases are mainly, or we can say that base arrangement same in all human, okay. And then, so yes, nucleotide bases as such mainly 3 into 10 raised to 9 base pairs, 99.9% bases are arranged same in all the human. And then secondly, as such, the gene based features as such. So total number of genes in the human genome, total number of genes somewhere around 30,000, but previously it has been estimated, it has been assumed that it may be 80,000 to 1,40,000, but in reality number of genes are only 30,000. So then one more important feature, average size of the gene, average size of the gene 3,000 bases, this much only. And the largest gene, yes, so codes for dystrophin, so dystrophin gene is largest. It has 2.4 millions of sequences, 2.4 million sequences or bases, okay. So that is the largest dystrophin. And then as such, the lowest number of genes on chromosome Y, okay, lowest genes on chromosome Y, it has hardly 231 genes, okay. And then when we talk about basically the highest number of genes on chromosome 1, that is 2,968 genes, okay. Yes, all these genes are the result of number of the bases. So number of bases as such, number of bases per genome, it can be 3,164.7 million. So this is what we can say gene based features of the mainly the human. So next, number 3, function based as such. We can say that 50% of the genes identified, functions unknown, and then only 2% of DNA codes for RNA. This is again function based, what we can say, the features. Then there is one more, what we can say, SNPs. Number 4. What is this SNPs? Single nucleotide polymorphism called as single nucleotide polymorphism. So these SNPs located at 1.4 million locations. So what is the use of SNPs? Presence of SNP make the DNA different from one another, make the DNA look different from one person to another person. Imagine here person 1, person 2, person 3. From a particular chromosome, we have taken their DNA. So at this location, same location, he has adenine, he has thymine, he has guanine. So with a single base, the DNA appears to be different. So thus with single base, the DNA become different from one another. Okay. So this is what we call as a single nucleotide polymorphism. So this is all about the features what we have discussed, mainly salient features as such. Nucleotide based approximately 3 to 10 raised to 9 base pairs are there. 99.9% basis arrangement very much same in all the person. And then the gene based total number of genes 30,000, but earlier it was estimated 80,000 to 1,40,000. Average size of the gene is minimum it will have 3,000 bases. Dystrophin is a protein, and that protein coding gene is the largest one, having the 2.4 million sequences. Lowest number of genes are seen in the smallest chromosome. Why? It has hardly 231 genes. Large size chromosome is number 1. It has highest number of genes, 2,968 genes. Number of bases of the genome in a per genome is 3,164.7 millions. Functional based, mainly 50% of the genes identified their function till not known. Only 2% of the genes or the DNA is functional, that form the RNA. And every human has at 1.4 million locations. They have their particular sequence of base, and they differ from person to person. Means how a person different from another person, because of 1.4 million location DNA is different. So this is all about what we have discussed, mainly the salient features of we called as mainly the Human Genome Project. What are the challenges and as such? Challenges mainly, we can say that the technology transfer was a challenge, storage of information was a challenge, then ethical legal issue addressing was a challenge. Okay, so number of the challenges it has been faced by mainly Human Genome Project. As such, understanding the chemistry of life, understanding the central dogma, understanding the expression of the mainly the genes was a challenge. So all these challenges have been overcome by mainly Human Genome Project as such. So Human Genome Project unwinded the entire human DNA and gave the lots of information related with the good genes, bad genes and gene defects. So this is what we talk about, mainly the challenges. You need to note down what is that. The challenge mainly as such, studying of the huge amount of the bases, 3 into 10 raised to 9, that means billions of bases. Second challenge, understanding mainly the chemistry of life, how the DNA, then RNA, how the protein, what is the genotype, what is the impact on the phenotype, understanding was a challenge. Then mainly we can say that coding itself was a challenge, and understanding the chemistry of life or metabolism was a challenge. Then preserving this information was a challenge. All these challenges have been encompassed by advancement of the technology. What are the technology used? They have used BAC, they have used YAC, they have used ESTs, they have used what you can say the automated sequencing method, then mainly PCR method. So all these methods helped mainly to overcome various kind of challenges. So with this, yes, we have completed mainly the concept as such human genome project. So in the next class, yes, we will be discussing about some more interesting concept. Till then, take care, study well. See you then. Bye.